We're now going to begin our study of living organisms by starting at the cell and working our way out. Chapter 4 actually has two parts. The first part is going to be your study on the individual organelles within the cell. The organelles are the small pieces parts within the cell, the nucleus, the mitochondria, and so on. You're going to research those and present them to me in a cell poster. This lecture is going to be the processes that occur within the cell, both of which will be Chapter 4. The first thing that we need to know as we study the cells is that there are two types, prokaryotes and eukaryotes. The major difference between the two is the number and type of organelles that are found within each. The second most important idea is the membrane. Prokaryotes do not have membranes surrounding these organelles. Eukaryotes do. On the test for the exam at the end of the year, and for my test, you're going to need to be able to compare the difference between prokaryotes and eukaryotes. So we'll start with the prokaryotes. First, the cell is filled with a gel-like material called cytosol. The cytosol does two things. First, it fills the cell and gives it support, and second, it gives a place for cellular processes to happen. Inside the cytosol are the organelles. The idea of cytosol and the organelles within it is called cytoplasm. So when you hear somebody talk about cytoplasm, they're talking about the organelles and the gel-like goo that they're floating in. The cytoplasm is one whole compartment. It's not divided up. So if you looked at it in our world, it would be a one-room apartment rather than a multiple-room apartment. Floating in that cytosol, you will find DNA. DNA is not within a membrane-bound nucleus. It is all by itself. And the DNA is circular. Also within the cytosol, you will find ribosomes for making proteins and a cytoskeleton for support. Surrounding the cell is a plasma membrane. Outside that plasma membrane is the cell wall. The cell wall gives the prokaryote extra strength. A eukaryote is very similar. It also is filled with cytosol, and the cytosol also has the organelles within it. It also has DNA, but this DNA is found inside a nucleus, and that nucleus has its own membrane. DNA in eukaryotes is called a linear form. We also know it as a double helix. The eukaryotes have all the organelles that a prokaryote has, plus many more. Eukaryote cells are what you're going to be talking about when you do your poster. Eukaryote animals have a plasma membrane surrounding their cells, and eukaryote plants have both the plasma membrane and the cell wall, very similar to what prokaryotes have. Another thing that's very important in eukaryotes is its endomembrane system. The endomembrane system is a series of membranes that divide the cytosol into individual compartments. Within each compartment, you have a different chemical process going on. So if you're in a prokaryote and it needs to make a new ribosome, that's all it can do at one time. It can't do anything else. If you're in a eukaryote, you can make a variety of different organelles all at the same time because each process is separated by a membrane. This allows eukaryotes to be more efficient. In addition, the ability to carry out several processes at once is a major factor in evolution. That's what allows eukaryotes to evolve into a variety of different organisms where prokaryotes' evolution is much more limited. This slide shows a comparison between prokaryotic and eukaryotic organisms. We're showing you on the left the archaea and the bacteria. These are the simplest organisms. Those are the prokaryotes. And you can see in the picture the different parts that we talked about. Because the DNA is not in a nucleus, they call it a nucleoid. The other thing I want you to see on this picture is the flagellum at the end. This is a long, whip-like cytoskeleton structure that allows the cell to move. Eukaryotic cells are the complex organisms, the plants and the animals. And again, this shows all the different organelles found within the cell, and you can see the membranes around each one. That membrane, as I said, is the major difference between these two cell types. One of the major questions that biologists have is, where did this first eukaryotic cell come from? Things don't just appear out of nowhere, they have to come from something else. So how did such a complex cell with all those different organelles come from a prokaryote cell which had such few organelles? They're still not really sure on all of the answers, but they think they might have begun to explain it using the endosymbiotic theory. This particular theory only explains how mitochondria and chloroplasts appear, but it works for these two organelles, and they're hoping that they'll be able to come up with something similar to explain the others. So I'm going to try to explain this endosymbiotic theory to you. I'm going to go through it step by step, but if you're still confused at the end, don't be too concerned, because I'm going to do it again in class, and I'm going to make it a little bit interactive so that you can kind of see what I'm talking about. So the first thing you need to know is we're going to go back millions upon millions upon millions of years ago. The atmosphere at that time was carbon dioxide and methane, other gases, very little oxygen. We had prokaryotes at the time, bacteria primarily, and they were anaerobic. Anaerobic means they breathe something other than oxygen. Over time, again millions of years, the atmosphere changed and more oxygen began to form. As the oxygen formed, aerobic or oxygen-breathing prokaryotic bacteria evolved. So now we've got two different types of bacteria, both breathing their own specific gas. Here's where we get kind of an iffy part. There's a process, they still don't really understand it, where the anaerobic bacteria, which were large, engulfed the aerobic bacteria, which were small, and brought them inside. They didn't eat them, so they somehow surrounded them, and the two began a symbiotic process, both of them alive and both of them using the other for survival. The aerobic bacteria was able to breathe the oxygen, so when the anaerobic bacteria floated through a patch of oxygen, it survived. As it floated out of that oxygen and into carbon dioxide, it could breathe, and the aerobic bacteria survived. So in both cases, it was a mutualistic symbiosis. They both were able to survive a poisonous gas because of the presence of the other. Over time, the aerobic bacteria adapted and evolved and became the mitochondria in animal cells and the chloroplasts in plant cells. So then your next question as a scientist is, this is a great theory, where's the proof? There are 4 bits of evidence. Number 1, mitochondria and chloroplasts have their own DNA, which is different from the DNA of the rest of the cell. 2, they have their own ribosomes. No other organelle has their own DNA. No other organelle has their own ribosomes. 3. They have 2 plasma membranes rather than 1. All other organelles have 1. Scientists believe the DNA, the ribosomes, and the membranes are all left over from when these organelles actually were bacteria. And the fourth thing is that mitochondria and chloroplasts are about the same size as a prokaryotic organism found today. So those 4 bits of proof seem to support the endosymbiotic theory. Once you have mitochondria or chloroplasts, we have the ability then to create new organelles and then to go from the prokaryote to the eukaryotic structure. We're going to focus now on eukaryotic cells. If you take a look at any size organism, whether we're looking at a whale or we're looking at a mouse, the size of their cells is approximately the same. You would think that as the organism gets larger, its cells would get larger, but that's not true. Cell size is actually limited, and here are the reasons process-wise. Okay, first, as cell size increases, diffusion rate increases, meaning that it takes longer for the materials that a cell needs to get in, and it takes longer for waste to get out. When you have a very, very large cell, diffusion rate could be so long that those vital nutrients can't get in, or waste products will build up too much and the cell will die. Larger cells need more energy for their cellular processes, just like a larger person needs to eat more to run a mile than a smaller person does. Larger cells need more materials for growth. If you're a larger cell, you've got a larger mitochondria, or you have more of them, so you actually need more materials to get things going when they're broken down or just to grow and function. And of course, a larger cell creates more waste, which means you have more energy spent in getting rid of waste products, energy that would be used for other cellular processes. As cells increase in size, they need more organelles to function. The more organelles that you need to support, to grow, to repair, the more energy and the more nutrients you need. If the cell gets too large, it simply can't get enough nutrients and enough energy to survive. As a cell's size increases, its volume increases more rapidly than its surface area. If you look at my 2 pictures at the bottom, you'll see 2 spheres of 2 different sizes. The little red line represents what I talked to you about before. The distance between the center, which would be the nucleus, and the plasma membrane, which would be the outside edge. As the sphere gets larger, that distance increases. For cells, that means diffusion time increases. Small cells have more surface area than large cells, so they are more efficient. More surface area exposes more of the cell's plasma membrane to the environment, allowing a more efficient exchange of nutrients in and waste out. This means that an organism made up of lots of small cells has an advantage over an organism made up of fewer larger cells. This is because more cells with more surface area means more movement of nutrients in and more movement of waste out. Everything happens faster, quicker, and more efficiently. There are some cellular processes that don't work if you have just more and more and more cells. So for example, a muscle cell. Muscle cells are very long and very large. They have to get electrical information back and forth along them so that they can contract and expand. And we'll talk about this quite a bit when we get to human systems. If they were a series of small cells, that movement of electricity wouldn't work as efficiently. But cell size is limited. We have to get those nutrients into the nucleus and those wastes out. Skeletal muscle gets around that by having more nucleus, more nuclei. And so you have a variety of nuclei all in one small area, and so diffusion, even though the cell is very long, is actually small because the nutrients only have to go a small distance to get to a nucleus. A neuron, which is the bottom picture, functions basically in the same way. In this case, we have one very long cell. Again, electrical impulses need to move back and forth along a neuron, and again, we'll talk about this when we get to human systems. It also must be one single cell because that electrical impulse will not move properly if it has to go through a long line of individual cells. A neuron gets around the size limitation by being long and very, very skinny. So again, that diffusion of nutrients into and out of the cell is very small. In this case, it's because the cell itself is very narrow, and the distance that those nutrients have to cross is also very small. Before I finish the idea of cell size limitation, I want to go back to the idea of diffusion and why that is so important to cell size. We've talked a little bit about that already when we talked about how surface area and diffusion are related. You increase surface area, you increase diffusion rate. We talked about it in terms of distance. If you decrease distance, you increase diffusion rate. Temperature we haven't talked about yet. If you increase temperature, you increase diffusion rate. However, we've also talked about how living organisms have a limited temperature range. So as you increase temperature, you increase diffusion rate. But you can only increase temperature so far before you begin to actually harm the organism. Concentration gradient can also be adjusted in order to change diffusion rate. A concentration gradient is the difference in the amount of molecules on one side of a structure or the other. So if you look at the picture, you see an artificial cell. There are molecules on the right. The membrane in the middle, it looks like a sheet of glass and a filter paper, and then molecules on the left. The concentration gradient in this picture is larger on the right than it is on the left because there are more molecules on the right than there are on the left. If you increase the concentration gradient, if you add even more molecules to the right side of this picture, it will diffuse into the cell even faster than it is currently. So like temperature, you can make diffusion rate increase by increasing this concentration. But like temperature, depending on what material you're increasing in concentration, you only have a limited amount before you actually harm the individual. The last thing I want to cover is the idea of apoptosis. Not all cells in our bodies are supposed to hang around forever. Some of them are actually supposed to die at a particular time. They're programmed into an organism's development by the DNA to die. That planned cell death is called apoptosis. Apoptosis isn't like accidental cell death, like an injury. So if you get a cut on your hand, those cells die. That's an accident. Apoptosis is timed to create a morphological change. Remember, morphological is a change in form or structure. So we want your cell to develop and to change, and that happens through apoptosis. So, for example, frogs, they begin as tadpoles, and through apoptosis their tail dies, those cells die, it disappears, and legs form. That's what I'm talking about when I'm talking about programmed cell death. The way it works is you have hormones or growth factors, chemicals that will start the cell to change. These signals, which are programmed in by the DNA, will begin the process of apoptosis. So what happens is the cell begins to shrink, the nucleus condenses, the DNA is broken into pieces. Nearby cells will engulf and digest that apoptotic cell by a process called phagocytosis. Phagocytosis literally means that a cell engulfs it and takes this other cell into itself and then digests it. Apoptosis is absolutely necessary for normal development and cell differentiation. If it doesn't have it, the organism will develop abnormalities or disease. The abnormality, for example, would be webbed fingers or webbed toes. When we are all a fetus, we all have our fingers webbed together like a frog. When that development has reached a point where the fingers can be separated, that webbing cell goes through apoptosis and it begins to die. So when normally it would be repaired and replaced, the body allows it to die. That allows that cell webbing to decrease slowly until we have separated fingers. This is the end of Chapter 4, Parts of the Cell.